Welcome back to Acing AP Biology. In this video, we will be going over sex chromosomes and genetic disorders. A heterogametic sex is a sex that produces two types of gametes and determines the sex of the offspring. A homogametic sex is a sex that produces one type of gamete. In human beings, males are heterogametic because they have both an X and Y sex chromosome. Females are homogametic because they consist of a pair of X chromosomes. The sex of the offspring between a male and a female depends on which chromosome the male donates. If the male donates an X chromosome, the zygote will be female. If the male donates a Y chromosome, the zygote will be male. The female parent can only put an X chromosome into the gamete. X chromosomes are very large, containing lots of genes. The Y chromosome is small. Sex-linked disorders take place due to defects in X-linked genes. These are most common in males because males can never be carriers as they only have one X chromosome, so the defective gene is always expressed. Females can be carriers because the dominant allele in one X chromosome can mask the defective recessive allele in the other X chromosome. Because of this, males are hemizygous, a term used to describe a diploid organism that contains only one copy of a particular gene. Fathers can pass down X-linked genes to daughters, not sons. Mothers can pass down X-linked genes to both sons and daughters. If a carrier mates with a normal individual, the defective gene will be passed down to 50% of the children. If a carrier mates with a male who expresses the defective gene, there is a 50% chance that each child will express the defective gene. Examples of sex-linked disorders include color blindness, Duchenne muscular dystrophy, and hemophilia. X inactivation occurs in female mammals. The XISI gene inactivates the genes in one X chromosome in every cell, condensing the X chromosome into a bar body. This inactivation occurs randomly and leads to diploid cells having only one functional X chromosome. This creates a mosaic, meaning that some cells are active with the maternal X chromosome and others are active with the paternal X chromosome. All mitotic descendants have the same X chromosome inactivated. For females, heterozygote for a sex-linked trait, half her cells will express one allele and the other half will express the second allele. The bar bodies are reactivated in ovaries so that the gametes produced all have an activated X chromosome. A good example of X inactivation occurs in the tortoiseshell cat. The tortoiseshell cat has a mosaic of black and orange fur due to X inactivation. As you can see, the cat is heterozygous for fur color, with one allele for orange fur and one allele for black fur. Then, due to X inactivation, the cat produces a tortoiseshell pattern, with some areas being black and some areas being orange. Next, let's move to our discussion of genetic disorders. I know we have talked about sex-linked disorders, but now we will go into other types of genetic anomalies. We will talk about two major categories: nondisjunction, which includes aneuploidy and polyploidy, and chromosomal permutations, like deletion, duplication, inversion, and translocation. Nondisjunction occurs due to an error in meiosis or mitosis, where homologous chromosomes or sister chromatids do not separate. This leads to an abnormal number of chromosomes in daughter cells. In this scenario, nondisjunction occurs in meiosis I. This leads to an extra chromosome in this cell and a missing chromosome in this cell. And then after meiosis II takes place, these two daughter gametes will have an extra chromosome, and these two daughter gametes will be missing a chromosome. In this scenario, nondisjunction occurs in meiosis II. So the two daughter cells produced from meiosis I have the normal amount of chromosomes. However, nondisjunction occurs in this daughter cell, leading to gametes that have either an extra chromosome or a missing chromosome. For this daughter cell, on the other hand, the two gametes produced are normal because nondisjunction does not take place. Aneuploidy is a condition where the individual has an abnormal number of chromosomes, resulting from nondisjunction. A trisomic cell has an extra chromosome. A monosomic cell is missing a chromosome. Down syndrome is also known as trisomy 21 because of the extra chromosome 21. Polyploidy involves a cell that has extra sets of chromosomes. Triploidy means that the cell has 3 sets of chromosomes, and thus they are haploid. Tetraploidy means that the cell has 4 sets of chromosomes. This can occur due to fertilization of an abnormal diploid egg produced by nondisjunction. Polyploidy is most common in plants. Chromosomal mutations are changes in chromosome structure. All 4 types affect phenotypic expression. These are potentially lethal. Crossing over errors can lead to deletions and duplications because DNA is not exchanged reciprocally. Deletion involves the removal of a chromosome segment. Duplication occurs with homologous chromosomes, where a deletion of a segment in one chromosome leads to a repeated segment in a homologous chromosome. Next come inversion and translocation. Inversion involves a segment that is reversed in chromosomes. The sequence of genes is therefore disordered. In translocation, a segment from one chromosome moves to another chromosome that is non-homologous to it. Basically, the 2 segments switch places. That's a wrap for Unit 10. If you have any questions, feel free to comment below. See you next time. For more bio content, hit subscribe.